All right, so what we'll talk about today is we're going to talk about important differences between hormonal and neural controls of the body. We'll talk about major endocrine organs, describe their locations. We'll do more of that in lab. We'll distinguish between what are called hormones, paracrines, and autocrines. We'll describe how hormones are classified chemically. We'll describe the two major mechanisms by which hormones bring about their effects on their target tissues. We'll identify factors that influence activation of a target cell by a hormone. We'll list the three kinds of interactions of different hormones acting on the same target cell. And then hormone regulation, you guys will actually be doing that for a post-lecture assignment today that I'll explain at the end of today's lecture. Okay, so the endocrine system, keyword being system, is one of the body's two major control systems. What is the other control system that you learned about last year in BIOL 32? The nervous system. Now, one thing you should also remember from BIOL 32 is the levels of structural organization. So I want you to take a second really quickly and try to put these in order from what you can remember from last semester or last year or whenever it was that you took BIOL 32. And we'll go from most simple to most complex. And why isn't my pen working? Okay, so from most simple to most complex, what is the first thing on this list? Should be chemicals. And then organelles. After that, and then organ systems, and then what's the last thing? Organisms. Okay. Hopefully that came back pretty good for you all. That's those are the levels of structural organization. We're going to do a review of that really quickly. And so you should remember that our chemicals include atoms and molecules. Molecules are when you have different elements that are put together. They're called compounds if the two elements are different from each other. But those are going to come together to make our organelles. Organelles are going to come together to make cells. Different types of cells that carry out the same function are going to come together to make tissues. Different tissue types that carry out the same function are going to come together to make the organs. Different organs with the same function are going to come together to make organ systems. And then all of those organ systems are going to come together to keep the function of the human body. So a little bit of a review. So organelles, of course, include things like mitochondria, ribosomes, nucleus, Golgi bodies, smooth ER, rough ER. We'll talk a little bit about those in today's lecture. And then when we get to tissues, what are the four major tissue types that you learned in BIOL 32? Epithelial is one. Connective. Muscles one. And then nervous. Those are your four main tissue types. Now you learn lots of specific tissue types, like simple squamous epithelium. That's a specific tissue type. But your four main tissue types or major tissue types are epithelial, nervous, muscle, and connective. Now the functions of the endocrine system include, it's going to be regulation or control of major body processes that include reproduction. Reproduction hormones include things like progesterone, estrogen, follicle-stimulating hormone, luteinizing hormone, but we're actually going to talk about all of those hormones at the very end of the semester. So we're not really going to get into reproductive hormones for this chapter, but we'll come back to it a little bit later. Endocrine system will regulate growth and development. We'll talk about growth hormone today. That's the major one. Also, it's going to control water balance. So we'll talk about hormones like antidiuretic hormone and aldosterone today and on Thursday. is going to be involved in electrolyte and nutrient balance. You did learn a little bit about a hormone that's involved in nutrient balance back in Bio 32. So in B32, you learned about calcium homeostasis. Does anybody remember the hormone that helps regulate levels of calcium? Now, acetylcholine, the hormone that parathyroid hormone. So parathyroid hormone is released when blood levels of calcium are too low. Okay. Does anybody remember the 3 targets for parathyroid hormone? How do we increase those levels of calcium? So small intestine 1 is going to trigger absorption at the small intestine. So we're going to take some of the calcium from the absorption at the small intestine. We're going to take some of the calcium from the food that we eat, and we're going to direct that towards the blood more so that we can bring those calcium levels up. What's another target for parathyroid hormone? Reabsorption of calcium at the kidneys. It's important that we say reabsorption. The calcium will be removed from the blood when we make urine, but then we're going to say, hey, we want to keep some of that calcium. Let's put it back into the blood. So we're reabsorbing that back into the blood. And does anybody remember the last target? Osteoclast activity. So we increase osteoclast activity. What do osteoclasts do? They break down the bone. So we get the osteoclasts to rev up their function, which is to break down bones, so that we can release more of that calcium from the bone into the blood. So all those 3 mechanisms, looking to the intestines, the kidneys, and the osteoclasts, that should bring our calcium levels back up to normal, and then we should have homeostasis again. Is everyone good with that? Okay. So we'll also use the endocrine system to regulate cellular metabolism and energy balance. We'll talk about thyroid hormone and metabolism, I think today. And then mobilization of body defenses. So we'll talk about one particular hormone that we won't talk a whole lot about, called thymosin. It helps us to develop our T cells. We'll talk about that later in the day. Any questions so far? Okay. So we'll talk a little bit about those 2 major control systems and what the differences are between the 2. One major difference is that the endocrine system is made from epithelial tissue, while the nervous system is obviously made from nervous tissue. How these cells of these systems communicate with each other and with the entire body. Actually, before we get there, I should have asked you. Do you guys remember what the specific tissue type is for glands that you learned in Bio 32? Yeah, it's simple cuboidal. So simple cuboidal epithelium is the specific tissue type that we have in our glands across the body. But how the cells communicate with each other in these 2 different systems is the endocrine system uses hormones via the blood in order to get a message sent to a distant tissue or cell. So an endocrine gland will release a hormone that travels through the blood to get to a target cell or tissue somewhere else in the body to get it to do whatever the desired response is. And so there is some kind of receptor specificity. So when hormones get released, they're only going to bind to a specific receptor on a specific cell. Okay? That's going to help us avoid any unwanted responses. We don't want hormones to just be able to bind to any cell in the body to carry out a specific response. We want it to go to a specific cell at a specific time. So there are receptors that are specific to that hormone, and that hormone won't bind to any other receptor. And then with the nervous system, they are pretty much physically connected. There is a microscopic space in between neurons or a neuron and a muscle when they are communicating with each other. So they're more physically connected versus hormones. They have to travel a long distance to the blood to get to whatever target cell they're trying to communicate with. So this is that receptor specificity. So here's the sending cell here, sending out the hormone. Now, this diagram refers to it as a ligand. Okay, a ligand is just a broad term for any chemical messenger. Okay, so we talked about in BIO32 ligand-gated channels where a chemical binds to a protein channel that allows it to open up. A ligand just means a chemical messenger. It's a broad term. So all hormones are ligands, but not all ligands are hormones. We'll talk about other different kinds of ligands a little bit later, but this is a broad term for a chemical messenger. So this could be a hormone. So it shows here's a target cell here, here's a non-target cell. So the sending cell is sending out a hormone or a ligand, okay, and it's going to bind to a very specific receptor. Okay? Any non-target cells that this hormone's not trying to communicate with, will not have a receptor for that particular hormone. Okay? So we'll only have hormones for specific, only receptors for specific hormones on the target cell that we want the response to happen. Questions about that? And then at any point, if you need me to slow down or need me to repeat something, please stop me. It doesn't make any sense for you to all be confused by yourself while I'm just talking up here. So if you need me to repeat something or if you have a question on something, please do not hesitate. Okay. So a little bit of a review on the nervous system. So make sure that you are familiar with the nervous system, watching on the back of the nervous system. Many times throughout the semester, we like to talk a lot about the sympathetic nervous system throughout the semester. If you remember, our central nervous system is where our brain and spinal cord is. Peripheral nervous system includes all the nerves that are going to communicate back and forth with the brain and spinal cord. And then we have two major divisions for function. We have the sensory division, and then we have the motor division. Sensory is to receive all information. The motor division is to get the parts of the body to do what we want them. And then two divisions of the motor division is the somatic nervous system and then the autonomic nervous system. Does anybody remember what is the general term for the, I guess I'll just say it. So effectors. We have effectors that we're trying to get to carry out a response, right, with the nervous system. What are the effectors for the autonomic nervous system? Do you remember? Glands is one of them. Not all muscles, but autonomic nervous system. Is that going to be voluntary or involuntary? Involuntary. So what are involuntary muscle tissues? Cardiac muscle is one of them. And then what is the other muscle that is involuntary? Smooth muscle. So our autonomic nervous system tells glands to release either hormones or other chemicals. Cardiac muscle to pump the heart, and then smooth muscle usually to get things going down the tract, whether it's urine or feces or a baby. Okay. And then what is the effector for our somatic nervous system, which is voluntary? It's voluntary. Somatic nervous system is voluntary. What kind of muscle? Skeletal muscle. So the effector for our somatic nervous system is skeletal muscle. And the somatic nervous system is voluntary. We tell our skeletal muscle when to contract. And then our autonomic is involuntary. Now, because the endocrine system uses hormones that travel a long distance throughout the blood, they do have a slower response time. So it's going to take a while for us to produce that hormone, export it out of the cell and the gland, get it to travel through the blood, and get it to bind to its target cell or target receptor. And so it's slow response, but it has longer lasting effects. So it may take a long time for it to happen, but it's really hard to shut off these responses once we have the hormone out there binding to cells and tissues. On the other hand, the nervous system, it is a fast response. It works off of electrical impulses, so that happens immediately, but the effects don't last as long. We have, what you should have learned in BIO32, is we have neurotransmitter termination. So there are ways to turn off neurotransmitters or stop them from working because we want that response to be fast. We don't want to have our muscles contract any longer than they need to. Does anybody remember the types of neurotransmitter termination that we have? What was it? ACH is the neurotransmitter, but what is the one that turns off ACH? Reuptake is one of them. So acetylcholinesterase. So acetylcholinesterase is the enzyme that breaks down acetylcholine to stop it from binding and stop it from working. So that's going to be called neurotransmitter degradation. So we just break apart the neurotransmitter so that it can't bind to the target anymore. And then the last one's they just diffuse away. Okay? Naturally, they'll diffuse from a concentrated area to a non-concentrated area, so they'll diffuse away from the synapse, and they won't be able to bind anymore. So this is a way. These are three ways in which we terminate neurotransmitter effects. By reuptake, so we recycle that neurotransmitter, put it back into the axon terminal where it came from. We break it down through an enzyme, like with acetylcholinesterase, or those neurotransmitters simply diffuse away from the synapse and can no longer bind. All right. Questions about the differences between the endocrine system and the nervous system? One thing you learned about BIOL 33, lots of things you learned about BIOL 33 that we're talking about today. You learned about the difference between an exocrine gland and an endocrine gland. It was during tissues that you learned it. Tissues 1, I think. So exocrine glands, they tend to secrete their products onto an internal or external surface. So it's usually in an internal or external passageway. Well, internal passageway or a external surface. And so an internal passageway, like your GI tract or your respiratory tract, some kind of tract in the body that has exposure to the outside. And then an external surface, and when it talks about external surface, it's usually talking about your skin. External glands include like your sweat glands that release sweat onto your skin. Does anybody remember the two types of sweat glands from BIOL 32? So the eccrine and the apocrine. Yeah. Or we have glands like your pancreas that releases enzymes into your digestive tract. That function would be an exocrine function. Now we'll talk about how the pancreas actually has endocrine function as well. It can do both. Okay. But an exocrine gland is going to secrete things onto a internal or external surface. So that means, because especially like with your apocrine and eccrine glands that are putting sweat onto the skin, we have to get that sweat from the gland to the skin. So exocrine glands tend to use ducts to get their secretions onto whatever surface they're trying to get it onto. And then endocrine glands do not use ducts. They are ductless. We'll talk about why in a second. Endocrine glands, they secrete their products or hormones into the bloodstream, and so they have direct access to capillaries. So there's no need for a duct. They're just going to release their products straight into the capillary. No need to take a tube and put it somewhere else when they have direct access to it. I'll show you the differences between those two. Anybody still need this up? Second, you don't need to do that because you're writing something down. Just tell me to stop and I'll take a drink of my water or coffee or whatever I have with me. Everybody good now? Okay. So, like I said, exocrine glands can put their products onto an internal or external surface. So they do use ducts. So you can see here's a little green duct here, here, here. So these are your salivary glands. Your salivary glands need to be able to release that saliva into your mouth. So they use little ducts to get it from the gland into the oral cavity. You don't need to know the names of those glands yet. You will when we get to the digestive system. But endocrine glands, however, again, have direct access to capillaries. So you'll see this thyroid gland here, this little butterfly or a bow tie looking guy. And he has direct access to capillaries. So he can just release those hormones straight into the bloodstream. No need for ducts or a exporting system. So again, here's more of at a cellular level, the difference. So and exporting glands would use a duct to release their product onto an internal or external surface, while endocrine cells have direct access to capillaries to just put that hormone directly in. So what is a hormone? A hormone is a chemical messenger or ligand that's produced by one set of cells that travel throughout the body via extracellular fluid, like the blood, or sometimes it can be lymph as well. It changes the metabolic activity of a different set of cells called our target cells. So one cell produces the hormone, releases that hormone for it to bind to a receptor on its target cell, a specific cell that it wants a response from. Any questions so far? That one? Okay. Okay. Anybody still need this up? Okay, so we're going to talk about now these chemicals here, these ligands. They're called autocrines and paracrines. Okay, they're talked about when we talk about hormones, but they are not considered actual hormones. These chemical messengers do not travel through the blood. Okay, so with autocrines and paracrines, these are ligands or chemical messengers. And if you go on your lecture notes, it says autocrines first, I think, then paracrines, and then it goes into what the difference is between autocrines and paracrines. This is the difference. So ligands or chemical messengers that release chemicals. Around their immediate space. So this is what makes autocrines and paracrines different from hormones, because they do not release their product into the blood. They're going to release their product right outside their own cells, and we'll talk about how they do that here in a second. So an autocrine is a chemical messenger that gets sent out by a cell, and then it binds directly to the same cell that released it. Okay, so it's a cell that's giving itself a signal to carry out a certain response. That's an autocrine. Auto means self. Okay, so this is a chemical that gets sent out by a cell and then binds to that same cell. Okay, this happens a lot in our immune system, where we have T cells that can direct themselves to make more copies of themselves whenever we have an infection. Right? That's an autocrine. A paracrine, you can think about it like a pair of cells, but these are chemicals that get released to a neighboring cell. So these are, again, chemicals that get released to a neighboring cell. So one cell sends out a signal and it binds to a cell that's right next to it. And this also happens a lot in our immune system. Sometimes we have a single cell that's infected by a virus, and it sends out a signal to get the cells next to it to produce antiviral so that they're not infected by that same virus. Okay? So that's a paracrine signaling that's happening. So these are different from hormones because they only are released in their immediate space right around their cells, not into the blood. Okay, but they work very similar to hormones. They're going to bind to a receptor on a target cell, but they just don't travel through the blood, so they're not considered hormones. So autocrines, okay, chemical messengers that exert their effects onto the same cell that secreted them. Examples include prostaglandins and smooth the muscle. The prostaglandins are a reproductive hormone that can get the male reproductive system to carry out responses, or like I said, the immune system, and I'll show you a picture of that here in a second. Our T cells use autocrine signaling, again, to tell themselves to make more copies of themselves when we have an infection so that we can fight it off faster. Also, there's something called programmed cell death, or also known as apoptosis. So sometimes when a cell is not functioning at the level that it should, that it can send a signal to itself to basically implode itself, okay, for it to die so that it's not causing any harm to the body. So here is an example of that autocrine signaling. So here is an activated T cell. It will send out what are called interleukins. We'll talk about that more when we get to the immune system. And it releases interleukins that then bind to itself so that it can then make more copies to fight off a particular infection. Okay, so that's autocrine signaling. Cells help itself what to do. Story message, chemical message. So questions about autocrine. So make sure that you know they're different from hormones because they do not travel through the blood. Endocrine signaling, again, is when a message is sent out to a neighboring cell. Again, it doesn't have to travel through the blood. It's being sent out to a cell that's right next to it. So signals produced locally and received by neighboring cells. They move by diffusion, and they are quick, short responses. Examples include clot formation. So we'll talk about that in the next slide. And then also synaptic signaling. So when a neuron talks to another neuron that's a neighboring cell, it releases a chemical that binds to a neighboring cell. That's also considered a form of paracrine signal. Any questions about autocrines versus paracrines? One thing to note while y'all are ready is that even though autocrines and paracrines are different from hormones, hormones can sometimes act as paracrines. So an example of that is going to be at the ovaries and testes. So the ovaries, they release hormones like estrogen, and sometimes estrogen is a trigger to allow for follicle and egg development. During that time, though, that would be considered paracrine because it's all happening within the ovaries. Okay, a set of cell releases estrogen that binds to another set of cells in the ovaries to get those eggs to grow and develop. That's paracrine. However, sometimes estrogen is used to create secondary sex characteristics. So it gives women butts and hips, gives women breasts and things like that. But that's where the estrogen being released either from, estrogen being released from the ovaries is then going to travel through the blood to the breast tissue, to the hips and buttocks, and that's where it's now working as a hormone because it has to travel a long distance to the blood to get the response to occur, okay, versus it happening straight within the ovaries. Same thing with the testes. Testosterone is used to trigger more sperm production. When it's doing that, though, that's paracrine signaling happening because it's all happening within the testes. Those are all neighboring cells. It does not have to travel through the blood and then come back to the testes. No, when we're talking about sperm production, that's paracrine signaling. But then when we have testosterone causing more skeletal muscle growth or causing the growth of the Adam's apple, more so in males than in females, that would be considered hormone signaling because it has to travel through the blood to get to those distant structures in order to cause that response to happen. Okay. Does that make sense? Sometimes it can act in both ways. So paracrine again, synapse or neuron to neuron is considered paracrine signaling because you have a single cell sending out a message where that message or ligand gets, or paracrine in this case, gets released by diffusion and moves to that receptor. Okay, and so that would be considered paracrine. Another example is blood clotting. So we'll learn when we get to blood vessels in a few weeks that our blood vessels have different layers. They have an epithelial layer on the very inside, then there's a smooth muscle layer after that, and then more epithelial tissue on the outside. So how the paracrine signaling works, and then I'll make our little, my different colors and my pins is not working on here for some reason, but we'll try to make it happen. So with our blood vessels, there is a layer of epithelial tissue, also a layer of smooth muscle. So during blood clotting, we have damage to a blood vessel. Usually it's the epithelial layer that gets damaged first. So what it's going to do when it gets damaged is going to release a paracrine and it's going to have that message bind to the smooth muscle right next to it. Okay? It's a different cell type, so this is considered paracrine signaling. Okay? So the epithelial cells get damaged. They release a chemical message that binds to the smooth muscle, and then the smooth muscle is then going to contract and do what are called vascular spasms to close that injury, okay, so that we can decrease the amount of blood loss for about 20 to 30 minutes while our clot forms and we get that tissue healed. That's considered paracrine signaling because the epithelial tissue is telling the smooth muscle tissue to then contract to minimize blood loss. Does that make sense? You don't have to know those steps yet. We'll get to it, but that's just an example. So the way the hormones work is going to depend on what they're made out of. We've learned or we're told many different times about 32 that structure determines function. Same thing with hormones. The way that hormone acts is going to depend on what it's made from. So we'll do a little bit of a review on biomolecules. So carbohydrates is one of our major biomolecules. Or macromolecules. What is the monomer for a carbohydrate? What's our single repeating unit for a carbohydrate? Monosaccharide. Now, what is the monomer for our protein? Amino acids. What about for nucleic acids? The nucleotide. And now lipids don't actually have a single repeating unit, but when they do break down, they're going to break down into a glycerol and some fatty acids. Does anybody remember the mechanism by which we put monosaccharides together or amino acids together, nucleotides together? Dehydration synthesis, right? So that's how we put those monomers together to create a, what's the plural of a monomer? When we put them together, it becomes a what? Polymer. Okay? What if we wanted to break down a polymer into its monomers? What mechanism would we use? Hydrolysis, right? Where we add a water molecule to break apart those bonds between the monomers. They're related, so we'll talk about that. What did I say? The structure of a hormone determines its. Let me go back so we can talk about that really quickly. There we go. So the structure of a hormone can determine its function, but it's also going to specifically it's going to determine how it gets transported, how long that response lasts, and also the types of receptors that it binds to. So we're going to talk about two major classes of hormones today, amino acid-based hormones and then lipid-based hormones. So because they're made from different molecules, okay, amino acids versus cholesterol and fatty acids, or we'll talk about cholesterol as well. But whether you are an amino acid-based hormone or a lipid-based hormone, that's going to determine how you're transported, how long your response lasts, and also the type of receptors that you bind to. And where those receptors are. A little bit of B33 review on cells. And structure determines function, and what a hormone is made from is going to determine how it acts. One of those types of hormones are amino acid-based hormones. We'll talk about amino acid versus lipid-based hormones. And for amino acid-based, we're going to talk a little bit about protein secretion and reviewing those concepts. So first, how is protein made? The central dogma of molecular biology is that all information or genetic information in the body goes through this pathway. So our genetic information goes off in our DNA. That gets converted into what next? Messenger RNA. And then that is used to produce what? Protein. Now, what is the name of the process that converts DNA into RNA? Transcription. And then how about the process that turns our mRNA into protein? Translation. Now, when we have the protein, then, does anybody remember what the name of the bond is that keeps all those amino acids together? Peptide bonds. They're held together by peptide bonds. Okay. And then next, where are proteins made? What is the organelle responsible for producing proteins? Ribosomes. Now we have two different kinds of ribosomes in the cell. We have what are called free ribosomes that are sort of just free floating in the cytosol. And then we also have attached ribosomes. And they're called attached ribosomes because they're attached to what? Either the reservoir, or sometimes they're also attached to the nuclear envelope as well. Now the difference between free versus attached ribosomes is that free ribosomes make proteins specifically to stay within the cell. Just internal proteins or enzymes that the cell is going to use itself. Okay? Attached ribosomes, though, they are going to follow that segregatory pathway for proteins that are either going to be for export or that are going to be embedded outside in the membrane. Okay? Free ribosomes are for making internal proteins that are going to stay inside the cell. and then attached ribosomes are going to make proteins for export or to be embedded in the plasma membrane. So like I said, the attached ribosome is going to be making ribosomes for export. Does anybody remember what the secretory pathway is? We start with the reservoir, then to the Golgi, then to a vesicle, and then exocytosis, yeah. I'll give you guys a couple minutes to write that down. Yes, ma'am. Yeah, so just be my voice and whatever goes on. So all these right now in our conversation is stop. There might be some cost, so you'll have to call on. Okay, anybody still need this stuff up? Now I do have a handful of lectures that are pre-recorded because I never taught this class online, it's always been in person. So like I have a couple of lectures that are pre-recorded that you can go review ahead of time. I think the immune system, lymphatic system, I think about 10 lectures throughout this semester are pre-recorded. If I ever have to miss a class because I'm sick or whatever, then I'll do the pre-recorded version of it. Yeah, the slides are already there for the entire semester. Yeah. And then really quickly, so we talked about ribosomes and the reservoir and the secretory pathway. But what is the other endoplasmic reticulum that we have? Smooth endoplasmic. This is a blackboard, so we just write extra notes that I can't fit on the slide. Yes. Oh, there we go. So functions of the smoothie are. Does anybody remember what the functions of the smoothie are? That's super way back, biology. What is it? Detoxification. So it does drug detoxification, and there's one other thing. It produces a specific kind of biomolecule, one of our biomolecules. Lipids. So smoothie R also does lipid production. So we said that we have two different types of hormones, amino acid-based hormones, and then we have lipid-based hormones. So our amino acid-based hormones are going to be made by ribosomes, okay, because ribosomes make protein. And then our lipid-based hormones are going to be mostly made by our smoothie R. Okay, so questions about a little bit of All right, so amino acid-based hormones, we'll talk about those first, and we'll talk about the lipid-based hormones. Amino acid-based hormones can be many different sizes. They can be as small as a single amino acid. They can be a short chain, which we call a peptide hormone, or they're going to be a full-fledged protein hormone, where they're very large and probably have a complicated folding pattern. So, thyroxine, which is our T4 thyroid hormone, and then triiodothyronine is our T3 thyroid hormone. You can just call them thyroid hormones together as a group, or you can refer to thyroxine is our most used one. So those, our thyroid hormones, are derived from a single amino acid. So they're very, very small. We'll talk about how they act based on that in a second. Oxytocin, which is the love hormone, it helps with breast milk letdown, also helps with orgasms, bonding, but that's going to be a peptide hormone because it's short chain. Versus insulin at the very top, which is a long chain made of like 51 amino acids. That's going to be a protein hormone. Okay, so hormones, amino acid-based hormones come in many different sizes. So amino acid-based hormones, because amino acids and proteins are water soluble, they're going to be water soluble, which means that they travel freely in fluid compartments. They have no trouble traveling in blood or cytosol or any fluid within the body. But can water-soluble products move across the cell membrane? No. So water-soluble, what is the word for water-loving? Hydrophilic. Now our plasma membrane that's made from a phospholipid bilayer, is that hydrophilic or hydrophobic? Hydrophobic. So that means, I'm going to do a little drawing with it. Now, when we say it's hydrophobic, the inside is hydrophobic. This is not the prettiest, and it's probably not super accurate, but I'm trying to do it quickly. Now, the inside here, our tails are hydrophobic. The phosphate heads are hydrophilic. But the inside is all hydrophobic. So if we have a amino acid-based hormone, which is hydrophilic, and it tries to cross the plasma membrane, once it gets to that bilayer, those hydrophobic tails, it's going to get kicked right out. Okay? Hydrophilic and hydrophobic do not mix, so hydrophilic molecules are not going to be able to cross that membrane. The hydrophobic tails are going to repel it and push it out. Okay. Does that make sense to everyone? Okay, so amino acid-based hormones cannot cross the plasma membrane. And then just a little note, whenever I make notes on here, once I take it off, I can go back to it. So everybody good here? Okay. Because when I do this, it goes away. Okay. So can it cross the plasma membrane? No, it cannot. These are generally fast-acting. We'll talk about why in a second, how they function once they bind to their target receptor. And because they can't cross the plasma membrane, they're going to bind to a receptor on the cell surface. So for these amino acid-based hormones, their receptor is going to be on the outside of the cell because they cannot cross and go into the cell. So when we talk about lipid-based hormones, though, they can actually cross and they'll bind to a receptor on the inside. But for amino acid-based hormones, they're going to bind on the outside. However, we do have an exception. Thyroid hormones here, thyroxine, T4 and also T3, because they're made from a single amino acid, they're very, very small. And so these ones only, they can't cross the plasma membrane and bind to a receptor on the inside of the cell. Okay, but that's only because they're very small. Okay, remember, things that can cross the membrane are things that are small, hydrophobic, and uncharged. Now, I know these are hydrophilic, but because they're so small, they are able to sneak their way past the plasma membrane and get to the inside of the cell. Okay, that's our one exception. And then a clinical application is a goiter. If you look at thyroid hormone, it's made from a lot of iodine. So we really need iodine in our diet in order to make our thyroid hormone, which is going to control our metabolism. And luckily for us, all of our salt here in the United States, for the most part, is iodized already. So it already has iodine in it, but make sure you get enough of that in your diet. Now, in other countries, iodine deficiencies are more common because their salts are not iodinized. So if you experience an iodine deficiency, what can happen is that your thyroid is going to overwork itself trying to make that thyroid hormone. Okay? But it can't because there's not enough iodine for it to do so. So what's going to happen is that thyroid gland is going to overwork itself, trying to find materials to make this hormone. You may experience what's called a goiter. And so a goiter is a swelling in the neck due to an iodine deficiency. Now, many people with goiters are then going to experience hypothyroidism, where their thyroid gland is not functioning at its best because it's not producing enough of that hormone, because it doesn't have enough of the materials to make that hormone. Now, the diagnosis for this particular person would be a goiter, because we don't really know what their metabolism function is by just looking at a picture, right? How many times having a goiter will lead to hypothyroidism? So any questions about goiters? We'll get to it. All right. So lipid-based hormones, these are not water soluble. Okay. Lipids are hydrophobic, right? So they're not water soluble. They do not like to travel in water. So many times when we have to export a lipid-based hormone, or really any lipid in the body, it's going to be bound to a protein, because proteins do like to travel in water. So many times it needs like a little buddy to take it where it needs to go, because it doesn't like water. So the receptors are for lipid-based hormones are inside the target cell, and why is that? Is because they're hydrophobic. Okay, so they're hydrophobic, meaning they can cross the plasma membrane. So they'll cross the plasma membrane, and they'll have a receptor waiting for them on the inside of that cell. And so how do target cells get receptors? We're going to talk about that on the previous slide. So the receptors are made from protein. Okay, protein comes from RNA. That RNA comes from DNA, right? And so how cells get their receptors is ultimately through genes. We have genes, which are short pieces of DNA that are going to code for a certain receptor. Okay. Now, if we're talking about a surface receptor, a receptor that's on the outside of the cell for a amino acid-based hormone, do you think a surface receptor is going to be made by a free ribosome or an attached ribosome? Attached ribosome, right? Because those are for export or the membrane. So surface receptors are going to be made by attached ribosomes. And then our lipid-based hormones are going to use internal receptors. So will those be made by a free or attached ribosome? Free, right? Because free ribosomes make proteins that are going to stay inside the cell. Our attached ribosomes make receptors for our amino acid-based hormones, while free ribosomes make internal receptors for our lipid-based hormones. Okay, so how lipid-based hormones work is that when the hormone binds to its receptor, what it's going to do is going to activate some genes. Okay, so the lipid-based hormone has a sort of longer process for carrying out its responses. It has to bind to a receptor. That receptor complex has to bind to a part of our DNA, and then get that DNA to undergo transcription into an RNA, and then translation into a protein, and usually that protein is an enzyme that's going to carry out whatever the desired function is. We'll talk more about that process here in a second. And then lipid-based hormones, because they do have this longer process where they activate genes and then trigger transcription translation, they're going to be slower acting, but it's harder to shut off those hormones. Okay, amino acid-based hormones, we'll talk about their method in a second. They work very, very fast. Okay, but they're easier to shut off, so those responses will work. Amino acid-based hormones, and here, I'll talk about this as well. I feel like I'm missing something on my slides, that's okay. So amino acid-based hormones, they are water soluble, and therefore they're cleared by our kidneys. So they don't stick around for very long because we're able to excrete them through our urine pretty fast. Okay? Versus lipid-based hormones, they are not water soluble. And then also we said before that lipid-based hormones, whenever they travel, they have to be bound to a protein. So that is going to be a really big complex of a lipid and a protein together. And so our kidneys can't clear lipid-based hormones, especially when they're attached to a protein. Because the lipid protein kind of complex is not going to be able to fit through the capillaries of our kidneys in order for our kidneys to clear them. Okay, so lipid-based hormones, because they are larger, they get cleared by our liver, which has much bigger capillaries with much bigger holes in them to be able to break those down and get rid of them. So lipid-based hormones are not water soluble, and these are cleared by our liver. So we get rid of amino acid-based hormones from the body a lot more quickly than we can lipid-based hormones. Breaking down hormones through our liver takes a lot longer than breaking down hormones or getting rid of hormones through our kidneys. And we'll talk more about their processes and why one is faster than the other. Okay, so if you are an amino acid-based hormone, you're going to work by what's called a second messenger system, which is a very fast system. Basically, how that works is in order for amino acid-based hormones to carry out their response, we already have all the materials needed to carry out that response and get something to happen. And then amino acid-based hormones, all they do is bind to a receptor, and then all those ready-made products just carry out that function. Okay? Versus a lipid-based hormone that has to cross the membrane, bind to a protein, get to a very specific part of the DNA, cause transcription, cause translation, and then that protein can carry out the response. That's a very much longer process than it is for the amino acid-based hormones. So our lipid-based hormones are going to be working through activating genes, which again is a very long process, or longer compared to the other. And again, we're going to be working off of very specific receptors. Our hormones are only going to bind to specific receptors on our target cells. What's up, Jim?
So how are the lipid hormones secreted?
Through our liver, which is a longer process. So a target cell response depends on the tissue type. Our tissues can only do what they are made to do. So muscle cells, when we want to respond to a muscle cell, we just increase or decrease contraction. Epithelial cells will increase or decrease the amount of whatever product they're supposed to secrete. And so tissues are going to act however they're supposed to act based off of these hormones that bind to them. So some common target cell responses is that it may cause ion channels to open or close, which may increase or decrease the plasma membrane potential. So you should have learned, and this is probably something that you may not have spent a whole lot of time studying in BIO 32, but I know it's in your notes. In BIO 32, there are things called EPSPs and IPSPs. EPSPs are excitatory post-active potentials. This makes it so that the cell is more excitatory and more likely to reach threshold, and so more likely to set an active potential. So sometimes hormones change the membrane potential, so that makes it more easy to carry out an active potential. So it makes things happen faster. Okay? Other times, hormones may cause inhibitory post-synaptic potentials. which takes that membrane potential down. It makes it harder to reach threshold, so it's harder to send an action potential, so things may slow down in that response. Okay. Or either make faster signals or slower signals. Sometimes hormones stimulate activity or synthesis of enzymes in target cells, so we have them make more enzymes. It may stimulate mitosis, like in the case of growth hormone, leading to repair, replacement, and growth of tissue. So there are lots of different types of responses, and we'll talk about all these specific responses for specific hormones as we go through. And that'll be more on Monday. So second messenger system. This is how our amino acid-based hormones work, okay? Those ones specifically. So there's going to be a kind of detailed process of how this works, but I want you, as far as like studying those and what you need to memorize, you guys can work off of the summary at the very top, okay? This is how amino acid-based hormones work. We first have the hormone, which is the first messenger. That's going to bind to a receptor. So you can see that here, those little pink circles at the top, and I'll actually do it on here so you'll know what I'm pointing at when you watch back the recording. So here's the hormone. It's going to bind to a receptor first. Okay, once it binds to that receptor, it's going to activate something called a G protein. Okay, that G protein, once it's activated, as the hormone binds to that receptor, that G protein activated is going to come over and it's going to bind to an enzyme. Once it binds to that enzyme, we're going to take some ATP and we're going to make something called CAMP. That stands for cyclic AMP, adenomonophosphate. Learned about ATP, adenotriphosphate. This is monophosphate. So, and then that CAMP, which is our second messenger, is then going to carry out the response. Okay. So again, a hormone binds to the receptor. The receptor is then going to activate this G protein. That G protein is then going to bind to an enzyme so that we can produce our second messenger. And the second messenger is what's going to carry out or trigger the desired response. So the advantage of this system is that it's a quick on and off. As all these things, these internal proteins, the G protein, the enzyme, the ATP to convert into CAMP, all that stuff is ready-made. So as soon as that hormone binds, all this gets carried out immediately. Okay? So the advantage is there's a quick on and off, okay? And that the internal ingredients are already made and ready to go as soon as that hormone lands. So this is why amino acid-based hormones work so fast, because everything's already ready to go once it binds. Okay? The disadvantage, though, is that if we wanted this response to keep happening, we would have to have repeated stimulation. You have to keep putting out hormones in order to get that response to keep going. So it's going to be energy intensive for the cell to make more hormones. So is everybody okay with that concept? Now, that CAMP, this is what's going to be, for the most part, our second messenger, but sometimes other chemicals can act as that second messenger, one of them being calcium. And we'll talk more about that during lab. But calcium is often used as a second messenger instead of that CAMP, which is why generally we like to keep our calcium levels low in the blood. It's only about 90 milligrams per deciliter in the blood, because we don't want any undesired responses to happen because we have too much calcium. We don't want calcium acting as a second messenger when we don't want it. Okay? But calcium can act as that second messenger in certain hormone situations, like oxytocin. Oxytocin specifically uses calcium as its second messenger. And then we do have some amino acid-based hormones like insulin who work on the cell membrane without the use of a second messenger at all. So there's always going to be a couple of exceptions, but that's the process that most of your amino acid-based hormones are going to use. So hormone to receptor to G protein to enzyme, and then lastly to that second messenger, which is going to carry out our response. So questions about that process? Okay. Now, lipid-based hormones, again, is a more drawn-out process. So here's our hormone here, and lipid-based and steroid-based are the same thing. Right? Steroids are a type of lipid, it's just in more specific versus general terms. You can use them interchangeably. So our lipid or steroid-based hormones First of all, you see it floating by itself. Most likely, it was just separated from its carrier protein. Because remember, lipid-based hormones or lipids don't like to travel in water. So most likely, right before this step here, it was dropped off by the protein that carried it here. Okay. So the steroid or lipid-based hormone, because it is hydrophobic, can cross over the membrane. And once it does, it's going to immediately bind to the receptor. So this orange thing here is the receptor. So it's then going to bind to its receptor. The receptor hormone complex is then going to enter the nucleus. It's going to bind to a very specific part of the DNA, a specific gene that it wants to activate in order to get it to undergo transcription into an mRNA and then translation to make a protein. And it's that protein that's carrying out the desired response, okay, whether it's an enzyme or some other chemical, usually an enzyme though. So again, that's how our lipid-based hormones work, or also our thyroxine, because remember thyroxine is made from a single amino acid. You can also call it thyroid hormone. But thyroid hormone is a single amino acid, so it can cross the membrane and acts just like the lipid-based hormones do. Okay. So again, it passes over the membrane, binds to a receptor. That receptor complex is going to bind to a specific gene or part of the DNA. That's going to trigger transcription into mRNA and then translation into a protein, and then the protein carries out the response. So any questions about that process? And that's really all we have time for today. So I will finish this lecture in lab, and then we'll do the lab during the time. And then what I'll do for the recording is I'll record in the first lab as well, and I'll put those two recordings together so you have the entire lecture. Okay, so like we mentioned, with lipid-based hormones, those hormones are going to come into their target cell because they can't cross the membrane because they're hydrophobic. And then they're going to bind to a receptor, create a receptor hormone complex. That's going to allow the hormone to come into the nucleus. And then once in the nucleus, that hormone is going to bind to a specific part of the DNA, a gene, that's then going to cause transcription into an mRNA and then cause translation of a new protein, which is usually going to be an enzyme that's going to carry out the desired response. And so this is just a little review about how transition works. So if you remember, our mRNA is going to have the code for the protein that we're trying to create. And then the mRNA is going to be organized in what are called codons. So codons are 3 nucleotides that are going to be specific to a specific amino acid. And so a tRNA with the anticodon that's carrying the amino acid is going to be added to this protein. Comes to the ribosome, binds to its codon, here's the anticodon, and it's going to have an amino acid that's going to contribute to that protein. Then we move over the ribosome, we go to the next codon, then a new tRNA is going to come with an anticodon and a new amino acid to bind over and over again until we make newly synthesized protein. Once we have the new protein, it's going to be folded. If you guys remember, there are levels of protein folding for protein. Anybody remember what the first level of protein folding is? Primary. And the primary is going to just be the sequence of amino acids. What's after primary? Secondary. And with secondary, that's what either the alpha helix or it could be a beta pleated sheet. And then after a secondary is tertiary. And then tertiary is all the bonds between the R groups of those amino acids. So we have some hydrogen bonding that occurs. We have some partial positive and partial negative interactions that occur, hydrophilic, hydrophobic interactions that occur. And so with all of these different interactions happening between all these different amino acids, it's going to cause a complex of really highly folded structure, creating that tertiary level. And then if you have a protein complex, there's actually one more level to that, which is called what? Quaternary. So this is when you have multiple amino acid sequences that are brought together to make a complex. This is when you have a protein complex. With multiple subunits. So this newly synthesized protein from this ribosome is then going to undergo its protein folding until it reaches its final conformational shape. And then that's going to be brought into a vesicle. That secretory vesicle is then going to transport that hormone to the Golgi body, from the Golgi body to another vesicle, and then from that vesicle on to the plasma membrane for exocytosis. All right, so questions about that? Everybody remember the protein folding and secretory pathway? Okay. Everybody still need these notes up. Okay. Now, with amino acid versus lipid-based hormones, I want you to know a little bit of a difference in timing. So amino acid-based hormones, they work over minutes to hours. So it takes minutes to hours to elicit a response when we're talking about amino acid-based hormones. We talked about how those hormones act faster. Okay? And we're talking about lipid hormones using this pathway here. This takes days to weeks. Okay? So amino acid-based response takes minutes to hours versus a lipid-based hormone response takes days to weeks. So some examples of that. And an amino acid-based hormone that I brought up earlier is insulin. Okay, insulin gets released when your when your blood sugar is too high. Okay, and the insulin tries to bring it down by causing us to take that glucose out of the blood, maybe store it in the liver, or maybe tell the cells, hey, there's extra glucose. Go ahead and use it for energy to carry out whatever functions that you're doing. So with insulin, you want that to work over minutes to hours. Okay, if your blood sugar spikes, you want to be able to bring that down pretty quickly. You don't want it to take days or weeks for your blood glucose levels to regulate. So insulin works off of it's an amino acid-based hormone, and there it takes minutes to hours. Or another amino acid-based hormone that was brought up is oxytocin. Oxytocin is the one that helps with labor and delivery. And so you want that to happen in minutes to hours, not days to weeks. You don't want to be in the delivery room for days to weeks trying to give birth to a baby. So that works off of minutes to hours. Your lipid-based hormones that work days to weeks are going to be things like your reproductive hormones. So like the hormones that are going to control your 30-day cycle as a female. And there's actually a couple of cycles. We'll talk about those later. But those lipid-based hormones are going to take a lot longer. Okay? But they also last a lot longer as well. So why are hormones released? To maintain homeostasis. This is a regulatory component of our body. It's used to maintain homeostasis and regulate our body functions. So homeostasis, you should worry about negative feedback back in bio-32. I know you did. So this is a negative feedback loop with the pancreas and insulin and glucagon. So you should have learned that a part of the negative feedback pathway is that you have a stimulus, okay, where there's going to be some kind of change in the body. In this case, we have increased blood glucose levels. Okay? Then there's going to be a receptor that's going to detect those changes. Okay? In this case, the receptor is going to be within the pancreas. The pancreas is going to be looking for changes in blood glucose levels. Okay? And that receptor is going to then send a message. Okay? So that receptor is going to cause the release of insulin. That insulin is then going to target two different set of cells. So when insulin gets released, it's going to tell our tissue cells, hey, there's extra glucose available. Go ahead and uptake it to use that for energy to carry out your function. Or also that insulin is going to tell the liver, hey, there's extra glucose. Can you store that in the form of glycogen so that we can remove it from the blood? Okay. So if the tissue cells are uptaking that glucose and your liver is uptaking that glucose to store it, those two methods, those two responses should bring our blood glucose down back to normal and back to home. Okay. On the flip side, sometimes our blood glucose levels drop. Sometimes you skip breakfast and it can make you a little weak. Okay. And so in this case, the receptor is still in the pancreas. The pancreas is going to be detecting changes in blood glucose levels. However, when blood glucose levels drop below our normal set point, we release a different hormone called glucagon. Okay, glucagon is easy to remember. That's when all the glucose goes. Okay, so glucagon gets released when our blood glucose levels drop. And then glucagon actually only has one target. It's going to target the liver and say, hey, can you take that stored glucose that we had before, and you're going to convert it back into glucose, release it back into the blood, and bring our levels down. This is negative feedback. We're out of range. We have receptors to detect that we're out of range. Then we're going to carry out a response to bring us back into our normal range. Not only do we have negative feedback from control, we have positive feedback. This is when we move further and further away from our set point on purpose in order to reach a sort of climactic event, okay, some kind of end result. Now, what are the three normal instances of positive feedback? Birth is one, sexual response, and blood clotting. Okay, so with blood clotting, we're releasing more and more platelets, more and more chemical messages, more than normal, out of our normal range until we reach a blood clot. Okay, we'll talk more about that process of blood. As far as childbirth goes, we're going to have increased signals being sent to the uterus to allow for contraction. You're going to have the uterus expand more than normal, more and more and more until we have childbirth, and then it'll all go back to normal. So with positive feedback, we're moving away from our set point to try to reach an end event. Versus negative feedback is as soon as we get out of that range, we're trying to bring it right back down. So we'll have three types of stimuli that we'll talk about that causes hormone release. That's going to be your homework. I'll kind of preview it a little bit, and then you're going to study it more at home as homework. So cells respond to a hormone if they have a receptor for that particular hormone. So we talked about that already. There's a receptor specificity. So a hormone is only going to be able to act on a receptor that's specific to it. Some receptors are expressed on the membrane of most of our body cells. So like thyroxine, which is our thyroid hormone, it controls metabolism. All of our cells have the ability to metabolize. So all of our cells are going to have a receptor for thyroid hormone. Okay. So some of our hormones have receptors all over the body versus some hormones only have receptors in very specific locations. So we do have a hormone released by our anterior pituitary called the adrenocorticotropic hormone. Okay? What that means is that its receptors are on the adrenal gland, but specifically at the cortex of the adrenal gland. Now, you guys learned about in BIOL 32, we have the cerebral cortex on the outside of the brain and then the cerebral medulla on the inside. So this just means that the receptors are going to be on the outside of the adrenal gland, very specific locations, versus thyroxine, which is body-wide because that's a function that happens body-wide. Adrenocorticotropic hormone is only for stress responses. Okay, so it doesn't need to be body-wide, especially if we want only that part of the adrenal gland to produce cortisol. Okay. So just know, sometimes we have receptors all over for particular hormones, sometimes in a very specific location. So target cell interaction with the hormone depends on three things. What would you think those three things are that are going to determine how big or small of a response we have with a hormone? First off, it's the amount of hormone that we just have available. How much circulating hormone do we have? Okay, so specifically our blood levels of that hormone. How much of that hormone is circulating at any one time? So that's going to influence our target cell interaction. If there's only a little bit of hormone circulating, we may only have a small response. But if there's lots of hormone binding to lots of receptors, we may have a larger response. So not only the amount of hormone, but also the amount of what is going to influence our interaction. The amount of strength, we'll talk about that next. Its affinity, we'll talk about that next. But also, what is the hormone going to bind to? Receptors. So it's going to be the number of receptors also that we have available. And the last thing is the affinity between the hormone and its target cell. So what that means is you should have learned in Bio 32 about the lock and key model when it comes to like an enzyme and a substrate, right? That a substrate binds to an enzyme. Now, sometimes the enzyme and substrate have a really strong affinity towards each other, and they can bind to each other really tightly, and it's hard to get those to come apart from each other. Same thing for hormones and receptors. There are some chemicals that have a stronger affinity or attraction towards each other, and so they bind more tightly to each other. So not only does the amount of hormone and the amount of receptors are going to influence our interaction with our target cell, but also the affinity between the hormone and the receptor. Because if it binds really tightly, then we're going to have a longer response because it's going to be harder to pull those two things apart from each other. And if their affinity is low, then it might just bind for a little, have a short response, and then separate. Right? So the affinity or the attraction between the hormone and the receptor is also going to have some influence on that response. Okay? Does that make sense? All right. So we talked about number of receptors. The number of receptors is going to influence our interaction between the hormone and the target. So there are times when our body can what's called downregulate or upregulate the number of receptors. So what downregulate means is sometimes if we don't want a large response or we don't want the hormone response to occur, we can downregulate or decrease the number of receptors available for that hormone. So one example of that with downregulation is right here. This is called GnRH, and we'll talk more about this on Monday. But this stands for gonadotropin releasing hormone. So gonadotropin releasing hormone is going to help regulate the amount of reproductive hormones that get produced and released. Okay, so the more times that GnRH binds to a GnRH receptor, the more sex hormones that we produce and release into the body. And so when sex hormones are high, or reproductive hormones, so when we have enough sex hormone circulating, then the body can downregulate the number of GnRH receptors. So if your sex hormone levels are high, and we don't need to go any higher, we have enough sex hormone, then we can decrease the number of receptors that we have for GnRH, so it binds less often, and so we're making less of those sex hormones, and we can kind of bring those levels down. Okay? Same thing goes for the opposite. If we have not enough sex hormone, then we can increase the number of GnRH GnRH receptors. So it binds more often, and then we make more sex hormone and bring those levels back up. So our body, our cells, have the ability to either put more receptors or remove receptors depending on what the needs are at that one particular time. So upregulation, the example it's giving is FSH. So with this here, there's like a string of reactions that occur. We're going to talk more about this in reproductive system at the very end of the lecture or end of the semester. But one complicated relationship is that estrogen triggers an upregulation of FSH receptors. So estrogen can cause our cells to increase the number of FSH receptors. FSH stands for follicle stimulating hormone. Follicle stimulating hormone. Follicle stimulating hormone, one of its functions is to stimulate the follicles within the ovaries to get egg development to occur. And so estrogen has the ability to upregulate FSH receptors. Then FSH actually stimulates estrogen production. And then FSH then upregulates LH receptors, which is another reproductive hormone called luteinizing hormone. And this leads to what's called an LH surge. We have lots of LH being produced, and that's directly going to cause ovulation. So sometimes our body does this on purpose in order to get a final event to occur. Okay, so we upregulate all these receptors so that we can produce more hormone. We cause a really big increase in what's called luteinizing hormone, and then that causes ovulation. Now, I don't need you to know all these steps, but don't even worry about that. But just know that there are times in the body where we're going to upregulate the number of receptors so that we have a larger response to lead to something else. In this case, ovulation. Okay. So our body has the ability to create more receptors or remove receptors depending on the level of the desired response. And that's what I need you to take away from this. We'll talk way more about these processes when we get to the end of the semester. Okay. So any questions about upregulation versus downregulation? Okay. So a review. Hormones circulating in the blood are either going to be amino acid based. If they're amino acid based, it means they're water soluble or hydrophilic. They are free form in fluid compartments, meaning they can float around on their own as they like because they are water soluble and they interact with water well. And they are removed quickly by the kidneys because they're small enough to fit through the capillaries of the kidneys and we can excrete them through our urine. But if you're not amino acid based, you're going to be lipid based, also sometimes known as steroid based. But lipid based are not water soluble. These guys are hydrophobic. And so because they don't like to travel in water, they're going to be bound to a carrier protein when they are in these fluid compartments, like in the blood or inside the cell. They are harder for the kidney to remove because they're so large and they're usually bound to a protein. And so most are removed slowly by the liver, which takes weeks to occur. Now, half-life. Half-life is the length of time for a hormone's blood level to decrease by half. Okay, that's what a half-life is. How long it takes for us to go from 100 milligrams of a particular hormone to 50 milligrams of a particular hormone in the body. Now, we can use both urine and blood tests to detect the presence of hormones. But a urine test usually tells us, because with urine, our hormones that are cleared by the kidneys happen pretty fast. So urine production happens pretty fast. So we usually use a urine test or urinalysis to test yes or no for the presence of a particular hormone or drug. Now, our Lipid-based hormones, those get cleared by the liver, which takes longer. So if you wanted to know exactly how much of a hormone that you have in the body, you want to do a blood test in order to look at actual hormone levels. And your liver and your spleen are the ones that are going to be detoxifying your blood and getting rid of things in your blood. So if you want to know the exact hormone levels, you would do a blood test. A urinalysis really is only going to tell you yes or no if the drug or hormone is present. Kind of the last thing that we're going to talk about together as a class is that there are 3 types of interactions that we're going to talk about, and these are interactions when 2 or more hormones are working on the same cell. So we have 2 different hormones that have the same target cell. So we'll talk about synergism first, because that's the easiest one. So synergism is when 2 hormones have the same target cell, and their combined effects amplify the response. Okay, make a larger response together. So with synergism, the combined effects are amplified. So one example is that both glucagon, which we just talked about, that gets released when our glucose is too low, glucagon and epinephrine, they both cause the liver to release glucose. So when our glucose levels are low, glucagon binds to the liver and tells the liver to take the stored glycogen, convert it into glucose, and release that glucose into the blood. Okay? And then epinephrine, when you're in your fight or flight mode, you want more glucose available so that you have more energy to either fight or flight from that situation. So when you have both of these hormones released at the same time, they're going to amplify that response of releasing glucose. And so together, they can cause the release of 150% of the glucose that either one of them can do on their own. So they both have the ability to cause an increase in blood glucose, but if they work together, that response is amplified by 150%. Okay. Kind of the opposite of that is something called antagonism. Antagonism is when 2 hormones have the opposite effect on the same target cell. Opposite effect on the same target cell. That example is insulin versus glucagon. So these are 2 different hormones. They both target the liver, but insulin tells the liver to store the glucose as glycogen, while glucagon tells the liver to break down that glycogen and release the glucose. So they do the opposite thing. They're both working on the same organ or the same set of cells, liver cells, but one tells it to store glucose, the other one tells it to release glucose. They work antagonistically to each other. And then permissiveness is more of a dependency, where one hormone's response depends on the presence of another hormone. So one example of that is that we talked about thyroid hormone. It is going to regulate your metabolism, so how fast are your cells working. So thyroid hormone has a permissive effect on your reproductive hormone. So thyroid hormone is needed in order for reproductive development of a fetus to occur on time. So, meaning if you do not have enough thyroid hormone being produced, reproductive development is going to be delayed, and you may have issues in that baby as they reach puberty. Okay? So that's a permissive effect. Or thyroid hormone needs to be present and be present at certain levels in order for other hormones to carry out their responses, which is reproductive development. Okay? Another example where thyroid hormone kind of plays a permissive effect is that thyroid hormone upregulates epinephrine receptors. And so without thyroid hormone, we would have a weak epinephrine response. So in order for us to have all the effects happen of our fight or flight response, so have our breathing increase, have our heart rate increase, have our skeletal muscles be able to contract more, we need thyroid hormone present. Okay, because thyroid hormone is going to create more receptors, and then if we have more receptors, epinephrine can bind more often, and we can have a bigger fight or flight response. Okay. So the response of epinephrine relies on or is dependent on thyroid hormone. That's a permissive effect. Okay. So questions about permissiveness versus synergism versus antagonism. So this is just when two hormones are acting on the same cell. They either have a combined effect, an opposite effect, or a dependency that occurs. And one needs to be present to one to critical thinking, because it's already 10:13, I'm going to have you take this home and think about it, and we'll talk about it on Monday. But here's a critical thinking question for you. So the female hormone estrogen is known to increase the growth rate of certain types of tumors involved in breast cancer, so it increases mitosis, allows the tumor to grow more, while other types of breast cancer do not respond to hormones at all. Think of some treatment options for these two different types of breast cancer. How would we treat a hormonal breast cancer versus a non-hormonal breast cancer? So think about that at home. We'll bring it up again in lab on Monday, and we'll talk about the answers and different treatments and what the best one is. And lastly, your post-lecture assignments. This is your first homework assignment. You're going to go in your textbook and learn about these three different stimuli. So if you look at your lecture notes, there should be, I think on page 12, it says post-lecture assignment 1 on hormonal stimulation. So in order for a hormone to be released, that gland needs to be stimulated to release that hormone. And there are three types of stimuli. We're either going to have a humoral stimulus. So what that means is that there is some kind of change in the blood composition that stimulates a hormone to be released. We already talked about one example of that, which was with the pancreas and insulin. So the pancreas is going to be detecting changes in blood glucose. If we drop or increase too much out of that range, then the pancreas is going to be stimulated to release either insulin or glucagon. That's a humoral stimulus. Next is a neurostimulus. So this would be like in the case of your fight or flight response. Okay, it's going to be a neural signal that's being sent. You get excited or embarrassed or afraid, and then that causes your adrenal gland to release more epinephrine. Okay, you'll get epinephrine released from the brain as well, but also from the adrenal gland. And that's going to be through a neural stimulus. Okay? And then the last one is a hormonal stimulus. We're going to talk more about this on Monday. where you have kind of a hormonal cascade. One hormone binds to a receptor on a cell that causes the release of another hormone. So one we're going to talk about is that the hypothalamus releases something called thyroid releasing hormone. When that gets released, it goes to the anterior pituitary and then causes the release of thyroid stimulating hormone, which then causes the release of thyroid hormone from the thyroid gland. That's a hormonal stimulation where one hormone is causing the release of the next hormone. So it's either a hormonal stimulus, a neural stimulus, or a humoral stimulus from changes in fluid. So any questions about those three? So go to your textbook or watch a YouTube video on those three different stimuli. Fill out the post-lecture assignment in your lecture notes, and then I'll check it on Monday. Questions?